Our next speaker is Sabrina Büttner from the University of Stockholm.
So, hi everybody. This is actually my first ARDD, and my thanks to the organizers for inviting me to this fantastic meeting. I'm really happy to be here today and to have the opportunity to share some of our research with you and also to listen to a lot of exciting talks. So in my lab, we are focusing on cellular aging and interorganellar connectivity, so the communication between different subcellular compartments, be it at membrane contact sites or via signal transduction pathways. Still, I thought that today, for this meeting, the project that might probably be of most interest for the audience would be a bit of a different one, and decided to talk about manganese-driven coenzyme Q deficiency. So an organism is born, it dies somewhere in between, it might reproduce and it will age, which is what we all are interested in. And we are using the unicellular model system budding yeast to understand cellular processes associated with aging. So what processes drive the aging, and in particular what compensatory mechanisms are in place to sustain viability when one subsystem fails. In extension, we're also doing Drosophila research to extend our finding from a unicellular organism to a multicellular one. We are doing chronological aging, and while probably most of you are familiar with that, I thought I'd give a very brief introduction. So what we do is we grow, is this the point? Here's the pointer. Oops, sorry, I messed this up. We grow yeast cells in liquid culture, where they happily divide and proliferate until at some point nutrients will run out and they will enter into a non-dividing stationary state where they will grow old until at some point they will die off. And this is just an example of how such a yeast lifespan can look like. So those cells are grown in standard glucose-based media where they will die off within 1 or 2 days. So very positively for us in the lab, it's a very short lifespan. If we now restrict, for instance, glucose, a regime known as caloric restriction, which everybody knows here will extend lifespan across species barriers, or at least health span, we can also extend the lifespan of our yeast cells. And more recently, we have added another longevity-inducing regime to the picture, which would be phosphate restriction, which very potently increases survival of yeast cells in stationary phase, mainly due to a strong induction of autophagy. So what we can do now is we can retrieve our mean lifespan, or we can take aliquots to perform some spotting assays. And as you can see here, for instance, for phosphate-restricted cells, they are perfectly able to re-enter the cell cycle and to start to proliferate again and form a colony while the control cells are dead. So this model is what we mostly use to understand how organellar function and organellar proteostasis, and in particular the wiring between those different subsystems, changes as cells age. But as I mentioned today, I would like to talk about cellular and organellar metal homeostasis during aging. So Jutta, a former PhD student and then postdoc in my group, was working on dysregulated calcium homeostasis during aging and how this would affect lifespan, but also interorganellar communication. And for that, she used cells that lack PMR1, which is a calcium manganese ATPase that pumps those ions into the secretory pathway. So just really briefly, something in respect to the functions of PMR1 and also the phenotypes associated with its dysfunction. So this is evolutionarily highly conserved and supplies the secretory pathway with both calcium and manganese. That means that this protein is on the one hand important to maintain proper levels of those ions in the secretory pathway for protein folding, processing, glycosylation, and so on. But on the other hand, is also important to maintain the cytosolic levels of those ions low. In humans, mutations of SPCA, so the human homolog, lead to Hailey-Hailey disease, which is a blistering skin disease that is thought to stem from disrupted calcium homeostasis, actually. And in mice, loss of SPCA leads to defects in brain development and early death. In addition, this ATPase has been connected with cellular demise associated with Parkinson's disease in different simple model systems. So the loss of PMR1 led to premature cell death during aging, but we were actually not able to connect this to dysregulated calcium homeostasis, though we tried hard. And then at some point, we finally got access to TXRF to do some comprehensive metal analysis in our biological samples. And what we found then was, yes, we expected that both calcium and manganese might be increased, but we were really excited to see that actually manganese is up to 10-fold increased upon loss of PMR1, and thought, why not using that as a genetic model for chronic manganese overexposure? And just to be sure that we picked the right model, we tested a lot of other deletion mutants, all lacking proteins of manganese transporters, buffering systems, and so on. But the PMR1 deletion mutant here to the right actually accumulated by far the highest levels of manganese. So we decided to go for that one. So manganese overload and manganese toxicity is actually highly relevant, both for agriculture as well as human health. Due to the high industrial use of manganese. It leads to bioenergetic failure across species, and in us it mainly accumulates in the brain, where it leads to neurotoxicity with symptoms that resemble actually Parkinson's disease. Interestingly, manganese overload is mostly associated with iron deficiency, which is thought to be due to shared uptake routes and shared transporters. Still, the molecular mechanisms, so the molecular targets of manganese toxicity remained completely unknown. So we decided to use our yeast model as a simple system to analyze and explore the molecular underpinnings of manganese toxicity. We started with a proteomic profiling of our wild-type versus delta PMR1 cells. And yes, the proteome was deregulated. GO-term analysis indicated that a process that was deregulated was cation transport, which was not that surprising because we deleted ion transporter. But what was exciting was that mitochondrial function seemed to be deregulated, both in respect to the TCA cycle as well as respiration. So what we did next is we checked mitochondrial functionality. And TXRF-based metal measurement in isolated mitochondria revealed that actually the cellular manganese overload translated into a hyperaccumulation of manganese within mitochondria. This was accompanied by a drop of mitochondrial transmembrane potential, reduced respiration measured as oxygen consumption, as well as the incapability of those cells to grow on media with only respiratory carbon sources, such as glycerol and ethanol. So yes, loss of PMR1 and the overaccumulation of manganese compromised mitochondrial energy metabolism. And I don't show the data here due to time limitation, but we actually had exactly the same findings when we treated our wild-type cells with high levels of manganese. So to identify modifiers of this respiratory deficiency induced by manganese overload, we went for a genome-wide high-copy suppressor screen and transformed our PMR1 deletion mutant with a plasmid library, a genome-wide plasmid library, and then we screened for restoration of this respiratory deficiency. And Jutta screened a lot of transformants. She actually retrieved 20 plasmids that efficiently restored respiration in those cells, but out of those, 19 coded for PMR1. But one is sufficient, right? And this one coded for COG7. So who is COG7? COG7 is a part of a multimeric protein complex within the mitochondria, which is termed CoQ synthome. This complex is evolutionarily conserved and is responsible for the biosynthesis of coenzyme Q, alias CoQ, alias ubiquinone. Probably all of you are familiar with this. This is only produced in mitochondria, is an important lipophilic electron carrier in the respiratory chain, but is also shuttled to all other cellular membranes where it functions as antioxidant. CoQ deficiencies are strongly associated with aging, but also different human diseases. And here just quickly the structure. So it carries a redox active ring as well as a lipophilic tail, and the length of the tail varies across species. And then we thought, okay, COX-7 could restore mitochondrial function. Could it also restore survival during aging? And yes, it did. So overexpression of COX-7 was sufficient to prevent the premature cell death of the PMR1 deletion mutant. And that was accompanied by a complete restoration of oxygen consumption. And again, just want to mention that environmentally induced manganese overload in wild-type cells gives exactly the same results. And then, of course, the next very obvious step was to measure the product of the CoQ syndrome, which would be coenzyme Q. And we did that in cooperation with Fabienne Pirel in Grenoble. And what we found was very encouraging. Actually, the deletion of PMR1, if you just focus on that one here, led to really a drastic drop in coenzyme Q levels. And again, same results were obtained upon challenging wild-type cells with high levels of manganese. We had a concentration-dependent drop of coenzyme Q. And what is very interesting is that we also found a very, very drastic accumulation of DMQ, which is a precursor of CoQ in the direct substrate of COX-7. So next, we turn towards the respiratory chain. And in collaboration with Martin Ott, we ticked the usual boxes. So we checked supercomplex assembly, but this assembly was fully functional. We also went for complex 3 and 4 activity, but also the activity was retained here. But electron transport via coenzyme Q to complex 3 was completely or almost completely absent. And simple addition of exogenous coenzyme Q to those isolated mitochondria was able to restore electron transport. So the only component in this whole system that was actually missing was really CoQ. And similarly, overexpression of COX-7 to supply endogenously CoQ restored the missing electron transport in our PMR1 deleted cells. So all of the data so far indicated that it's really selectively the COX-7 catalyzed step that is impaired when cells are challenged with manganese. So, of course, we ask what's wrong with COX-7 when manganese is high. We checked transcription was unaffected. We checked mitochondrial protein import was unaffected, but still the COX-7 protein within mitochondria was almost completely absent. So we thought that maybe this is due to premature proteolytic removal and went for the major protease within mitochondria. It's the lone protease. In yeast, this is called PIM1 and generated a strain that allows us conditional depletion of PIM1. And indeed, what we found was that the levels of COX-7 in the PMR1 deletion mutant could be restored to almost wild-type levels if we depleted PIM1. So yes, this was obviously due to proteolysis. Why? If you remember from the beginning, I think I had it on one slide, COX-7 is actually a di-iron enzyme. And it's assumed that those two iron ions are incorporated into the active site while the protein is folded within the mitochondrial matrix. So we thought that it could be that the high manganese pool might somehow interfere with this and might cause a mismethylation of COX-7. And to test this hypothesis, Jutta established a setup where she could purify COX-7 in sufficient quantity to afterwards go for TXRF-based metal determination in a purified protein. And what she found was that indeed, when she purified COX-7 from PMR1 deleted cells or from wild-type cells challenged with high manganese, the manganese to iron ratio was increased. And this came along with a drop in hydroxylase activity of COX-7 measured in vitro with a purified protein. So if only COX-7 is impaired during manganese toxicity, then we should be able to circumvent this when we feed with a CoQ precursor that does not require this hydroxylase activity. And we did that. So DHB is a precursor that already carries the hydroxyl group that otherwise would be added by COX-7. And indeed, when we supplement our PMR1 deletion mutant with this DHB, we were able to restore respiratory growth and thus oxidative phosphorylation. So obvious question, of course, phylogenetic conservation. So to test that, we used the other model system we have available in our lab, which would be the fly. And this work has now been done by Philomena, a postdoc, and our Drosophilist in the group. We decided to go for muscle-specific downregulation of SPOC, so the Drosophila homologue of PMR1, because it's a mitochondria-rich tissue and a very energy-demanding tissue. And she assessed larval to adult developmental time and found that the depletion of SPOC causes a developmental delay that is accompanied by compromised mitochondrial function, visible here in larval muscle tissue stained with mitotracker to visualize mitochondrial transmembrane potential. And again, this was accompanied by reduced CoQ levels. And lastly, this developmental defect could be overcome by supplementing with DHB, so the head group precursor that bypasses the COX-7 catalyzed reaction. So to sum this up, cellular overaccumulation of manganese translates into a hyperaccumulation of manganese within mitochondria, where it will interfere with correct methylation of COX-7, leading to its inactivation and premature degradation. This in turn then inhibits CoQ synthesis and leads to bioenergetic failure that drives premature cell death during aging in yeast and causes developmental defects in Drosophila. Right now we're working on translating what we've done during Drosophila development into aging, and it looks very promising. And actually it's rather unknown whether mismethylation plays a big part in nature, but about 1/3 of all enzymes are actually metalloenzymes. And while a few of them have dedicated chaperone machineries or biosynthetic machineries that generate the cofactor, mostly those metalloenzymes only rely on a balanced metal pool for correct methylation. Thus, it will actually be important to understand how cellular and organelle metal pools change with stress and change with age. And the last slide, just to come back to the manganese-induced inhibition of CoQ synthesis. This was, of course, upon challenging cells with manganese. So is this relevant during physiological aging as well? I do not know yet, but we have first data that indicate that in the fly brain, manganese actually accumulates with age. We know CoQ levels drop with age. Whether this is somehow related to this increase in manganese levels is, of course, unclear, but we are following that up right now. And with that, I would like to close with big thanks to my awesome team that make science fun each day. In particular, Philo, Lucas, and Jutta were involved in this work, and then great collaboration partners, Martin und Nora Vögtle and Fabienne contributed to the study I just showed you. And then, of course, thanks to my funders. Without them, no research would be possible. And thanks to you for your attention.
Thanks for a great talk. We have time for one question. Where's the microphone?
All right, I will ask a question. Maybe I missed it, but could there be a competition between manganese and other cations in like calcium transport or other types of that could be outside of this CoQ degradation area?
Yeah, for sure. So you're asking for other reasons that might explain our findings. So for sure there is competition for transporters and uptake routes between different ions, and most transporters are not specific for one. So I think there are other reasons for the defects we observe upon manganese overload than COX-7, but they are what our data indicates are also related to mismethylation. So those di-iron enzymes, COX-7 is of course not the only one. There are others, for instance, the ribonucleotide reductase, so a prime target for chemotherapy, is also di-iron enzyme. And those enzymes seem to be prime targets for mismethylation. So it seems that the manganese and the iron flip-flop quite easily in those enzymes. Yeah.
Thanks again.